Good morning, Advanced Kentucky AP Science or AP Biology students and a few teachers out there I see. I'm thrilled that we have such a good turnout for this morning's content review session. We are lucky to have Robert Bauer presenting on cellular energetics. Robert's from Barren County High School and has been to the AP reading many times, and he knows a thing or two, so listen to his words of wisdom. I'm Lou Akampora, the Director of Science, and I'll be hosting, assisted by Monique Rice and Sherry McGuffin. There are quite a few students here, so let's please mute your microphones, turn off your video, please, and but do use the chat box if you have questions or comments, and Robert may ask you to type in some questions, so don't be shy. This meeting will be archived at kyap2020.com, along with a very nice set of notes that Robert has prepared for you. So once again, this is Robert, and I'm going to turn the meeting over to him.
All right, thank you, Lou. Share screen here real quick. All right, Lou, we're good to go. We see the screen okay?
I think we're good to go.
Okay. Looks like we've got several people here today, excited to be with you guys today. We've got an hour or so to go over cellular energy. This is something that probably takes a good couple of weeks in class with some labs to go with it. So we're going to move very fast, but if you have questions, put those in the chat box for me. We'll try to get to those and address those as we go. And we've got several questions to ask you along the way. We're going to focus a little bit on essays. So hopefully we can have a very productive hour. I just wanted to throw this little slide in there. Obviously, as you know, we're taking the test on May 18th. But if College Board needs to know if you do not have the technology to do that. Obviously, most of you are on Zoom today, do have that technology. But if you don't, please go to that website and make sure that everybody has that technology to complete the exam. All right, so we'll open it up with this. Over here on the right side, I have just made copies of, basically this is what the curriculum framework says. So as teachers, this is what we're supposed to teach you. So the reason I put that over there was for you to see exactly what the wording is. And if you feel comfortable with what that says, there's a really good chance that you're going to feel comfortable on the essays that they're able to give you. So we're going to start out with something very easy. And we all know that all living systems require a constant input of energy. That's why we eat. That's why we probably eaten a whole lot during this quarantine. But what would happen if, for instance, this system was not getting an input of energy, let's say? An open system is a system in which we have a constant input of energy, and therefore in this system right here, it's never going to reach equilibrium because we have a constant input, a constant output of energy. So as long as we receive an input of energy, we're continually going to have that water flowing down to turn that turbine there, if you will. But if we have a closed system where we don't continually put energy in, it eventually reaches equilibrium. And equilibrium is a word maybe you associate with something that's good. A lot of times we say equilibrium and homeostasis are the same thing. Well, we probably shouldn't associate those two things. Equilibrium, when we get to that balance, it's going to be a bad thing for us in biology. If you get to equilibrium because of a closed system, it's going to eventually equal death for the organism. So we have to have energy. And that's why this is an important couple of chapters that we usually have. So everything has to come in. We have to have more energy, obviously, coming in than we have going out for us to maintain an open system and maintain life. Okay, so something really easy to get us started. We'll continue with ATP. We all know that's our short-term energy release molecule. So we've got millions and millions of these made every second, every minute. Let's start off real quick in that chat box with this first question. We know what ATP is possibly made up of. It has those three parts. What ATP would be an example of what kind of macromolecule? Just throw that one in there real quick in the chat box. Just a quick question. We'll see what you guys come up with. Just give you a few seconds to throw that one in there. See a lot of different answers here. One of almost every kind. We got carbs, we got proteins, we got nucleic acids, we got everything except a lipid. So the correct answer here would be, this would be an example of a nucleic acid. The singular thing would be a nucleic acid, or this would be a nucleotide. So a nucleotide is made up of 3 parts. It's made of a phosphate group, it's made of a pentose sugar, and it's made of a nitrogenous base. The other 2 examples that we commonly say nucleic acids would be what? What are the 2 quick examples of nucleic acids that we would also say besides ATP? So very quickly we see those right answers. Yes, DNA and RNA would be correct. That might be something to throw in your pocket and remember. It might come in handy very quickly. The 3 parts of DNA, RNA, and ATP are going to be phosphates, are going to be sugars, are going to be that nitrogenous base. The only difference is obviously there's 3 phosphate groups for ATP and 2 for ADP. So we'll close that chat group out there real quick. So how do we make ATP, and why do we need to continually have this cycle here that we call phosphorylation? We go from ADP to ATP, back and forth, back and forth. So if you add a P, that's usually called phosphorylation because we add that phosphate group. So ADP has less energy than the ATP. What is this? I know you went over this in the last couple of weeks with your previous sessions. When I add water to that ATP, it breaks that down and becomes ATP. So we're releasing that energy. What is that process called when we add water to break a bond? All right, a lot of correct answers. So the word for break would be lysis. And if we're doing that with water, that's going to be hydrolysis, or obviously hydrolysis. The other option would be down here. When we go back from ADP to ATP, we're going to have to add a bond or add that 3rd phosphate, which gives us that energy. So it's the breaking and building. The breaking that bond releases energy. The storing of energy is in building that bond or putting that bond back together. So obviously, synthesis means to make, so making a bond or storing that energy would be on this side or the left side of this graph. So that would be dehydration synthesis. So very, very good on the chat box. Thanks for all your participation. 90 people in here, so I appreciate you participating and doing that very quickly so we can get as much covered as we can. Okay, so we'll look down here. There's 2 types of reactions, an exergonic and endergonic reactions that we kind of talk about. So this is going to be my release of energy. I know it's a release of energy because my products have less than my reactants. This is an endergonic reaction where I take in and store energy. So reactants have less energy than the products. We're going uphill. There's a positive amount of change here. This is a non-spontaneous reaction. So the change, if you look at the y-axis here, so over time, the free energy is going to increase. So that free energy would be the dependent variable in that graph. Reactants increase as they are put together to make the product. So that's a positive change in energy, a positive free energy change, and this is a negative free energy change that happens spontaneously. You should associate that a little bit with enzymes too. You've seen this graph very commonly with enzymes. A lot of times there's this little heel here. Let's see if I can draw on there. A lot of times you would have a little heel here that you would have to overcome, and that little gap right there that I have would be called what? I know that's review from your last session that you had. What's that little gap called right here that has to get that reaction started? Throw that in the chat box for me real quick. So that's called your activation energy. That's the energy that's needed to get started. Very good so far. So your activation energy, we try to lower that with enzymes, and that's obviously part of the energy unit too, but not our focus today. So exergonic is going to release that energy, and endergonic is going to take in energy. So Obviously, our focus today is going to be on cellular respiration and photosynthesis. So let's see if I can do this real quick. Here's your reactions, photosynthesis and cellular respiration. We know those, and we're pretty familiar and comfortable with those. But let's go back a slide and look at this. So hold on, try to go back. There we go. Okay, so exergonic reactions. Would cellular respiration or photosynthesis be an example of an exergonic reaction? So let me say that again. Would cellular respiration or photosynthesis be, hold on a second, Lou, not yet. This is just a chat box type here. We'll get some polling questions here going in just a second. Would the reactants lose energy as they become products? So would that be an example of cellular respiration or photosynthesis? Type that one in the chat box for me. You can just put CR or P to save time here very quickly. Yeah, we got a ton of CRs there. So cellular respiration. So that would mean we release energy in cellular respiration, and that would obviously, the opposite means here we would store energy here in photosynthesis. So let's see if that makes sense in our equations that we have real quick. So look at where we're going right here. So this last sentence I think is very, very important. A product of a reaction in a metabolic pathway. So metabolic pathways would be your sum total energy. So metabolism is that building and that breaking, otherwise known as the breaking would be a catabolic reaction, and the building would be an anabolic reaction. I know you went over those in the last couple of weeks too. So a product of a reaction in a metabolic pathway is generally the reactant of another pathway. So look at this real quick. This is a very, very straightforward part of these two processes. So let's draw on here real quick. So you've got the products of photosynthesis are the reactants of cellular respiration. And then the same thing, the products of cellular respiration are the reactants of photosynthesis. So because of that fact right there, what do we call those two reactions? What relationship do those two reactions have right there? That's another chat box real quick for me. You guys are really good on this. You're getting on this really quickly. I very much appreciate it. I see inverse, I see coupled. So here's what I see inverse a ton. I would prefer the term coupled is what I would. So because why would we call them coupled? If we go back to the slide that we looked at a while ago, if we go back to the slide we looked at, can endergonic reactions happen without exergonic reactions or vice versa? They can't. So those two reactions are dependent upon one another, or they're coupled reactions is a better way maybe to say that. So I see some inverse, I see some coupled. So let's just kind of think of it as coupled because one can't happen without the other. And that goes back to that previous slide that I had at the very beginning of the presentation, where we have to have a constant input of energy. Okay. So how are we doing so far? Everybody tracking and following along? Hopefully. All right, so there's the coupled part of that again. There's the products of chloroplast, or products of what we have in photosynthesis, or oxygen and glucose obviously go into that mitochondria, and those become water and carbon dioxide. Obviously, we produce the ATP as a byproduct of that. Okay. Any questions so far? We feel like we're tracking okay. I don't see any questions maybe in the chat box, but if you have those, you can obviously throw those in there and we'll get them answered. Okay. All right. So, all right. Good. Thanks, Austin, for that thumbs up there. Appreciate that. All right. So I know you've seen this, and we're not going to spend a ton of time on this, but here's our two questions. This is what you're tested over this year. These are your essays. We know that question 1 is going to be a 25-minute question. You have 25 minutes to do that, then 5 minutes to upload it. And then you can move forward to question 2. You've got 15 minutes to do it, 5 minutes to upload it. 65%, 35% of your exam for your 100% total. Just a couple of asides real quick that you might put in the back of your mind and remember. If we were doing this on paper, we would be able to move back and forth between question 1 and 2. Not this year. You're going to have to do all of question 1, submit it, and then move on to question 2 and submit it before your time runs out. So you've got to get those questions submitted before that time runs out so they have a chance to be able to be graded. If you wait and try to use extra time, try to circumvent the system, if you will, you'll get a 0. They won't be able to score it. So it's very important that you log on early, you get situated early, and you're ready to go on that test day to be able to answer the questions to your best of your ability. What I want to focus on this slide, just very, very quickly, is this right here. We know that these words, we know describe, identify, justify, analyze, make, and justify. We know those are all going to show up in this question. This is going to be an 8 to 10 point question. So we know we have to get some of those points based on the fact that that's a 65% of our exam to be able to pass that. We come down here, we do the same thing. We look and see that we have describe, we have explain, we have predict. All of those are going to, and then the last one would be support. Those are all going to be, sorry, jumped ahead. Let's go back there. Hold on. There we go. Sorry about that. So we know those are all going to be in that short answer question. So that short answer question that we have right here has those 4 words, and we know that describe, explain, predict, support are all going to be 1-point questions. So we know for a fact that there's 4 points there in that question. So that's going to be your short answer up here at the top. That's going to be your long answer. So I just wanted to make sure you saw that. I know your teachers and you're going to get a chance to practice that on your mock exam. So if there's any questions about that, throw those in the chat box and I'll do my best to answer those. But that's just kind of your introduction to that. You're going to have to focus on knowing what these words mean and exactly what to do to satisfactorily answer the question. One thing you notice is not on there. There's not going to be a graphing question. You're not going to have to make a graph. I know some of you, maybe you were worried about trying to upload that or draw a graph or any of those things. You will not have to do that, but there's a chance, a very, very, very good chance that you're going to have to analyze some graphs and figure out is there statistical differences or what those graphs look like. So it's going to be the platform the exam is going to be on. It's going to be completely online. You're going to log on. They're going to send you a link to log on to that. And it's going to be where you can completely type your answer or you're going to be able to write your answer and then upload it. So there's not going to be any graphs that you're going to have to put on there. You could do it all typing if that makes sense. But that doesn't mean there won't be calculations. So I know we're diverted a little bit, but I think that's clearly important for you to be able to understand what you're going to be tested over. And that's why I wanted to look at that real quick before we did this question. Okay, so we've got a couple of polls that we can throw up here in just a second after we look at this question. So we've got our introduction. We have a word identify here. We have explain. We have another identify. So based on this, this is clearly a short answer question. There's a 1 point here, there's 2 points here, and there's 3 points here. So I've got 3 questions on the poll that I would like for you to fill out, and let's see what we've got. So matter continuously cycles in an ecosystem. So based on that, based on the fact that that matter is continuously cycling in the ecosystem, what is number 1? What is number 2? What reaction would be number 1? We might want to scoot that over the side so we can still see the question. go ahead and put your answers in that, and I'll give you 30 seconds or so to do that.
So students should be able to select answers for questions 1, 2, and 3 on the poll, and that poll box can be scooted off to the side.
Okay. Can you see that now? Is that okay?
I don't see any polling votes yet.
So CO2 to organic molecules to CO2, what would we identify? Identify is just, you're going to have to tell me what it is. So is it going to be cellular respiration or photosynthesis? And same way, what is it for 2? So you're identifying the key metabolic process for step 1 and the key metabolic process for step 2. So Lou, I don't know, we don't have any results yet in that. Okay, she's saying it doesn't let you submit without doing all the questions. So just do those first 3 and hit submit. How about that? Just do those first 3 and hit submit. Can we do that? Or is it you have to answer all of them? Okay. So Lou, that might not work very well. You're gonna have to answer all of them before you submit it. How about let's just do this, guys? How about let's just change the game real quick and let's just do this. Type it in the chat box. Type it in the chat box. We'll take that poll down. Type it in the chat box. So what's the key metabolic process for step 1? If we're taking CO2 and we're making it into organic molecules, what's that going to be? Is it going to be cellular respiration or photosynthesis? Just type it in the chat box for me. How about that? Overwhelmingly, right? Photosynthesis. So that would make number 2 what? That would make number 2, obviously, cellular respiration, right? Okay. Thanks for doing that. Sorry the poll didn't work out very well, but you guys are obviously 100% polling on here. So that shows me that you know what you're talking about and doing that well. So if we look at those equations, for each process, explain what? Explain how carbon moves throughout the cycle. So we've got CO2 going to an organic molecule. What's that organic molecule going to be? Specifically, it's going to be glucose, right? So and then if we come down here, that glucose is then broken down and we release it back out of CO2. An organism that carries out both processes, the choices were, here's the choices. How about you just type in which one of those it would be? In the chat box, just type in what would be an organism that does both of those processes, cellular respiration and photosynthesis. You can type A, B, C, or D in there for short. Okay, go. So everybody's throwing oak tree in there. Plants, oak tree, yeah, all those are correct. So if you think about it, obviously chloroplast and mitochondria is not correct because what? Those are not organisms. But a plant would have a chloroplast and mitochondria to be able to do both of those processes. A trout would obviously be able to only go through cellular respiration. Okay, so that just kind of is a warm up to get us started. And you see what a good answer looks like. You know, this guy didn't use, or this girl didn't use more than 10 lines here to give me a really good 3-point answer. So, you know, we're not trying to get you to write a ton, but we're just trying to get you to make sure you use those identifiers. So let's move forward a little bit and see if we can get to the meat of all of this real quick. So last question, and then we'll move forward to photosynthesis and cellular respiration. Identify one inorganic source of new plant mass and connect it to the cellular process underlying the increase in mass. So let's look at this real quick. To explain the increase in the mass of light-grown plants. Identify one inorganic source of new plant mass. So here's another word, identify. We obviously are going to have to do that. That should be just a very simple, we know what it is. So without me giving you choices this time, identify one inorganic source of new mass in this one right here. So it says the ones grown in, I'm sorry. There we go. So in number one, we have no treatment. So that's probably our control. Number two, grown in light. So in treatment group number two, what would account for it going up by 18 grams? What would be an inorganic source that it makes it go up by 18 grams in the week? So throw those in the chat box real quick. We got to move quickly. All right, so there's two correct answers to this. Light obviously is going to use two reactants. Those two reactants would be water and CO2. Light is a driving force that makes it happen, but that light is not going to allow us to gain any mass. It's going to catalyze the reaction, if you will. So that CO2 is a correct answer and water is a correct answer. Conversely, what would explain the decrease in mass if a plant's grown in the dark? Conversely, to explain the decrease in mass, identify the overall chemical reaction that is occurring. What is the overall chemical reaction occurring in treatment group number 3? Go. So it's cellular respiration, right? Cellular respiration, when a plant is placed in the dark, when it doesn't have light to drive photosynthesis, it's got to obviously use that energy that it made. It's going to use that glucose, and it's going to break down that glucose in the process of cellular respiration because that plant needs ATP too. So that goes back and re-emphasizes a plant goes through both processes, photosynthesis and cellular respiration. So hopefully we've got that basis to move forward to this next slide here. All right, so here's photosynthesis. We've got about probably 15 minutes or so to go over both photosynthesis and cellular respiration. So 15 minutes on photosynthesis and close to that on cellular respiration. So, all right. So photosynthesis captures energy from the sun and produces sugars. All right. Photosynthesis happens first in prokaryotic organisms, and then it obviously moves to eukaryotic organisms. So how do we capture energy from the sun? That CO2 is going to be reduced. What does reduced mean? If we think about our biology, or maybe more so chemistry, we think about this mnemonic right here. And that would be Leo says Gur, and this isn't a chemistry class, so we're going to kind of just use the basis of that to understand what we've got. That loss of electrons is oxidation. So that water is losing that hydrogen, which also has electrons on it, to become oxygen. And that CO2 is gaining those electrons to become a glucose. So that is why it is reduced. So very important concept of oxidation and reduction that we'll see as we go throughout here. So we have two parts, the light reaction and don't say the dark reaction, it's the Calvin cycle. The light reaction obviously happens in the light, and the Calvin cycle is independent of light. So you might hear that it's light independent reaction sometimes. So let me get this back on here. Okay, so your light reactions happen in the thylakoid membrane. So they're going to take in the light. That's why this is the photo part of the reaction. Lost my cursor for some reason. Hold on just a second. should have that back. Okay. So the light reaction, the photo part is happening in light, where that light hits that water and it's going to, like we said a while ago, it's going to oxidize that. So that means it's going to break off or pop off those hydrogens and therefore electrons. It's going to use that light energy in the electron transport chain in photosystem 2 and photosystem 1 to make me ATP and NADPH. That energy, because this happens with no light or independent of light, the Calvin cycle needs the energy we make in the light reaction to drive the making of, or the synthesis of, CO2 into a sucrose. So the light reaction is where we get the photo part. The synthesis is where we get the sugar. So that's just in summary, that's where you get photosynthesis. Okay. So make that a little bit more complicated here. That's just a quick summary. To make it a little bit more complicated, obviously the pigment that we get to absorb that light is chlorophyll. So chlorophyll absorbs the light energy. It boosts those electrons up. So when you move those electrons up, we get them to a higher excited state. And then we use that potential energy to move those electrons down the electron transport chain. So photosystem 1 and photosystem 2 is embedded within this thylakoid membrane. So this is the thylakoid membrane. The thylakoids obviously are stacked in grana here. Those thylakoid membranes are going to be within the chloroplast. The outside or the liquid part is called the stroma. So the light dependent reaction happens on the thylakoid. The light independent reaction happens in the stroma. So this is a very quick, what happens, photolysis happens to start this. So the photon, or the packet of light, is absorbed by that chlorophyll. The chlorophyll energizes that electron to a higher energy state, and then we use that potential energy to make ATP and NADPH. So this is what a photosystem looks like. This is clearly where it happens in the chloroplast. So let's break that down just a little bit, looking at the photosystems. So we're going to try to create a gradient. We're going to try to create a proton gradient to power the making of ATP and power the making of NADPH. So how do we do that? All right, so we bring in one of our reactants is water. So water obviously is going to be photolysed, and what that the light, lyse means to break. So we're going to break that water and we're going to change it into oxygen. What we get off are the hydrogens and the electrons. The electrons are sent into the electron transport chain, and the hydrogens are used to establish that proton gradient that we've got over here. So what we're doing, we're going to send those electrons, we're going to boost them up. That makes them have more energy. They're going to be taken by these proteins and moved down the chain. So look at this. The first one, as we move down the chain right here, first one as we move down the chain, it's going to go from a higher energy level to a low energy level. So that means it's losing energy. But what we use that energy for is to pump. We're going to pump hydrogens. And when I say the word pump, you automatically should trigger what in your head? What should be triggered in your head when I say pump? Throw that in the chat box. Yeah, active transport, right? So active transport, use of energy, any of those things would be the things that I would automatically come up with. So we have a low concentration here, a high concentration here. When I move from low to high, I'm going to have active transport, or I'm going to have energy. So I'm pumping, actively pumping, using energy from those electrons to get those hydrogens over here. So let's go ahead and make ATP. So we pump it from a low to high, and we now have established a proton gradient. All right, so that the hydrogens come from the water, they're pumped across, and then when they go down here, what is this? ACE means what? What does ACE mean? ACE automatically should be associated with, yep, very good. Thanks for jumping on there quick, enzymes. So this is an enzyme that synthesizes ATP. So this is my ATP synthase. Now I have a high concentration to a low concentration, so I do not have to have any more energy. This thing spins right here and phosphorylates ADP into ATP. All right, here's your hard question for the day. What is, or how is this ATP made? What process actually makes this ATP? So I'm looking for a very specific term here of how we put together that ATP. Let's see if who gets the bonus point. There we go, Trevor. Trevor's the first one to get that one. And then I'm sure others were just a little slower typing, but Sarah's got the right answer too, Taylor. So it's photophosphorylation. It's photophosphorylation specifically. Okay, so we've got phosphorylation because we're adding a phosphate to that ADP to ATP, but we used it from the energy of this light originally. So that's why it's called photophosphorylation. So we've got one of our intermediates, which is ATP. Now we've got to make the other one. But the problem is we're out of energy. You see how that's dropped down here. So we have to use the photosystem 1 to excite those electrons again. We're using the same electrons. We can reuse them just like we reuse ATP or ADP back and cycle it to ATP all the time. So we're energizing it again. So photosystem 1 energizes it, and then it drops down and makes me something called NADPH. NADPH, we're going to add a hydrogen. When we add an electron, when we add a hydrogen, what was that called again? It was called, Leo says GER. So the GER, when we gain an electron, we're reduced. So this is, again, an enzyme that reduces NADP plus to NADPH. So very simply, we've used the power of that light to energize electrons and make two intermediates, ATP and NADPH, and we can send those to the Calvin cycle and power those. So I like this little analogy here. You got the photon, it energizes it, drops down, makes ATP, re-energizes it, and makes NADPH. So the only thing here, photosystem 2 obviously comes before photosystem 1. That's not so obvious because you would think we'd go numerically, but photosystem 2 was discovered second. Photosystem 2 was discovered first. They just figured out what order they went in after they'd already named them. All right, anything on questions here? No, it just looks like everybody's responding there. Okay, terrific, terrific. All right, so that's the first. Now we need to move very quickly through. You see a ton of stuff right here in this Calvin cycle or light independent reaction, but I want you to know 3 steps very simply. We have to make sure we understand that the light reactions are going to make ATP and NADPH, and then that power is going to be used to produce carbohydrates from carbon dioxide. That's all we need to know, and that happens in the stroma of the chloroplast. So if you're very comfortable with what that says, you should be very comfortable with the Calvin cycle. So we're going to fix carbon. Carbon fixation is the first step. So what is that carbon that carbon dioxide is the carbon that we're going to fix. We're going to fix that with an enzyme called ribisco. Ribisco is the most abundant enzyme in the world. Think about how much photosynthesis happens. So carbon fixation, we use ribisco to fix that into a molecule of RUBP. All right. So that's carbon fixation. Then we rearrange that using ATP, using NADPH, and we make it into a molecule called G3P or PGAL. PGAL is a 3-carbon sugar. Our problem here is carbon dioxide is a carbohydrate is a 6-carbon sugar. So that means we have to go through this process twice to get me a carbohydrate. So that's what that says down here at the bottom. Okay. So I don't want you to get caught up on all these intermediates. I don't want you to get caught up on the big words here. I think you just need to make sure you understand that carbon dioxide is fixed using an enzyme. We take it through some different intermediate steps using ATP, using NADPH to power that into overall carbohydrate. We regenerate some RUBP so we can do that again. That's why it's called a cycle. All right, that takes us through photosynthesis. Any questions on photosynthesis? You can throw those in the chat box. I'll power on here. We'll look at this real quickly and then we've got to go probably. Okay. So we have light, we have dark, and we have boiled. So we know if we go back here, we know these are proteins, right? Protein, 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 all these are proteins. And if we boil something, proteins are probably going to be what? What would happen to those proteins? Throw that in the chat box real quick. They're going to be denatured, right? Denatured just means they're going to change their shape, and we have to have a specific active site. So if we change the shape of that active site, then we've got problems. More specifically, it's not going to work. If we put something in the dark, we're not going to be able to obviously have the first step, which is the light reaction. So the light and the unboiled here would have a change in transmittance. And the transmittance means that this DPIP, which is an electron acceptor, changes from blue to clear. If it changes from blue to clear, that's basically saying that photosynthesis is working. So this one right here is the only one we have a change in. So it's changing from blue to clear. So it's telling us that photosynthesis is working. It's not working here because obviously we have to have the light to make ATP and NADPH. It is not working here because we boiled it, which probably denatured those proteins. So I do want to touch on one very, very, very quick thing because I think we could see this. A null hypothesis basically states that everything would be the same. An experimental hypothesis is what we're used to making. That's saying that this one, sample one, is probably going to show a change in transmittance because, and you can fill in the blanks there like we just did. A null hypothesis would simply state that all three of these should have no difference. So there should be no difference in the percent transmittance of sample 1, 2, and 3 because that null hypothesis would generate no difference or no, we could say no difference between the different samples. So a null hypothesis is meant to say that everything is going to be the same or there's no discrimination between those different samples. Okay. We're going to skip over that one real quick so we can get the cellular respiration so we've got enough time to make sure we have questions at the end. Okay. So again, this is my guidance that we use to teach you all. It looks like a ton, but when we break it down, it's not really that bad. It's not really that bad. So this is my summary of cellular respiration. Obviously, cellular respiration is going to be the opposite of photosynthesis because now we've got that glucose that we made, and now we can break it down with the hopes that we're going to get ATP. So we oxidize, lose electrons. So technically, we're losing that H, and that becomes CO2. This oxygen gains the H or gains the electrons, so it is reduced. So that shift becomes how we make ATP, okay? So what I want to show you is how do we break down that glucose into intermediate steps and actually use that to make ATP? We're going to have to first, in the cytoplasm or the cytosol, we're going to have to take glucose and turn it into pyruvate. So that's what glycolysis means. So glyco is the glucose, lysis is we just break it in half. So this is a 6-carbon. We're going to change that into pyruvate, which is 3 carbon. So literally, we break that 6-carbon molecule into 2 3-carbon molecules. All right. So during that, we produce something called NADH. NADH is going to be an electron carrier. So when we reduce it, we add a hydrogen. If we say hydrogen, I'm going to say electron acceptor. But you can also say energy carrier. So those NADs and those FADs are going to be something that it's actually going to transfer energy as we move through. So let's look at this, and then we'll go to the next slide, which breaks it down a little bit. So glycolysis goes into the mitochondria. If we have oxygen, it's turned into acetyl-CoA, which goes into the citric acid cycle. Citric acid cycle produces a bunch of NADs and FADs that we use in oxidative phosphorylation to make ATP. So let's break that down for you a little bit. Okay, glycolysis is our first step. If the ins and outs make sense to you, you should be good to go. So glycolysis is a pathway that releases energy in the form of ATP. It releases NADH, and it releases pyruvate. So let's see where those three things come from. So glycolysis, remember 6-carbon, we break it down into 2 3-carbon molecules. We produce some NADH and ATP. So just a quick question here, that obviously happens in the cytosol, but where is the majority of the energy right now? Where is the majority of the energy right now in my output? After we've gone through the process of glycolysis, where's the majority of energy? Obviously, we've got some energy we can make in ATP. That's the only usable energy we have right here is ATP. And we actually net 2 because we had to put 2 in to get it going. So we had your activation energy technically was 2 ATPs, if you want to think of that. So the majority of the energy, let's see what you say, pyruvate, excellent, that's right. So the majority of energy is in pyruvate, and if we are in an aerobic process, so if we have oxygen, we can go on into the mitochondria. If we do not have oxygen, we have to go down here to fermentation. There's two types of fermentation. Obviously, a very lucrative process in the state of Kentucky is ethanol fermentation. And in our bodies, we have lactic acid fermentation. So the reason that we have fermentation, I said pyruvate has the majority of energy. NADH, we cannot use, ATP we can. So the reason that we have fermentation is to be able to continue. So those NADHs are actually broken back down. So we take that H off. So if we lose electrons, we oxidize those and we go back into a NAD so we can start the process over again when we do have oxygen. So basically the reason that fermentation happens is not necessarily to make lactate, it's not necessarily to make ethanol, but it's actually to re-get these starters or these inputs that we can use the next time glycolysis comes around, when hopefully we're in an anaerobic situation. Or I'm sorry, an aerobic situation, not anaerobic, okay? So let's go over here real quick. I went ahead and put the Krebs cycle on here. So pyruvate goes into this little transport protein is like a gatekeeper. If we have oxygen, we're able to move through that. If we're able to move through that, then we can continue on. If we do not move through that, that's obviously when we have to come back down here and go through fermentation. So that pyruvate is broken into something called acetyl-CoA. That acetyl-CoA is taken into the citric acid cycle, or the Krebs cycle, you might have heard. The citric acid cycle is mainly breaking that pyruvate into acetyl-CoA into these NADs and FADs. So what we have, the majority of my energy now is in NADs and FADs. Have we produced some along the way energy? Yeah, there's an ATP right there. We can actually use that, but we've only produced like 4 or 6 or so ATPs. We can't really use that. That's not really helping us a ton. That's not much different than fermentation down here. So the NADs and FADs are where the majority of my energy is, and we're gonna have to use that. So the NADs and FADs are taken to the electron transport chain, which is in the inner mitochondrial membrane, right? So last little section here. So we've got 10 NADs, we've got 2 FADs. So we're putting all that in. We had to use oxygen to get here. So what are my outputs gonna be? I'm hopefully gonna make a ton of ATP through this. So let's see how we do that. So NADH is taken in by, this is 1, Q, 2, 3, all those are proteins. All those are proteins. This is electron transport chain, just like we saw with photosynthesis. So that NADH is converted to NAD+. So we talked about this 10 times already. When I take an H off, what is that called? What is that called? When I take that H off, throw that in the chat box real quick. Called oxidation, right? Thanks for throwing that in there, oxidation. So remember, this is called oxidative phosphorylation. So this is called oxidative phosphorylation. So the oxidative part comes from when we take that H off, when we take this H off and make this an FAD or an NAD+. So we're using that energy to pump hydrogens, to pump hydrogens, to pump hydrogens, right? So if we're pumping hydrogens, that means we're moving from a low area here of hydrogens to a high area of hydrogens here, again, producing a proton gradient. Well, look, we use that energy 1, 2, 3 to pump those hydrogens across. When they run out of energy, those hydrogens and electrons come down here and jump on oxygen. Oxygen's your final electron acceptor to make water. So we get water as one of our products. But the most important thing is here is this chemiosmosis part. So the electron transport chain is the oxidative part because we're oxidating NADs and FADs, all right? The chemiosmosis part is obviously going to be the phosphorylation part. So phosphorylation changes obviously ADP into ATP using ATP synthase to make ATP. So we built these intermediates, NADH, in the in the two previous sections in glycolysis, in the Krebs cycle, but we get the yield of the ATPs when we actually take it through the electron transport chain and chemiosmosis. Okay, we won't read all this over there, but there's a ton of stuff that basically summarizes what I just talked about in that section. Okay, all right, so we're getting close to the end. So does anybody have any questions about cellular respiration that they can throw in the chat box? If so, we can answer those. If not, we'll finish with a couple of quick essay type questions. All right, I really like this one. This one is a quick and easy one. This would be a short answer type question. So I'm going to give you some data, and what you have to understand is you're not going to get a question that just asks about cellular respiration. You're not going to get a question that just asks about photosynthesis. You're going to get a question that asks you to integrate multiple units. So we talked about this unit specifically, but there's also this unit, which would be about unit 6, messenger RNA expression level. So that, how do we make messenger RNA? I know you talked about that last week. Messenger RNA is made through the process called what? You have to go from DNA to messenger RNA, and that's made through what process real quick? When I pop up here, I should see a bunch of transcriptions, right? So the thought is when you get messenger RNA, that if you're going to use the energy to transcribe, but you're also going to translate that too. So that means that gene would be expressed. So this gives you the tissue, this gives you the different genes, and it's asking you this. Based on the data provided, which is obviously here, we've got another identify. Identify the gene that's most likely to encode a protein that is an essential part of glycolysis. So there's going to be reading up here. I didn't put that reading up here just for time's sake, because you can answer this without. You can answer this without the reading. Some will answer this, or hopefully you can answer this in the chat box. Based on the data provided, identify the gene. So it's either going to be E, F, G, H, or I. You're going to identify the gene that's most likely to encode a protein that is an essential part of glycolysis. We'll worry about the reasoning here in just a second. Okay, so throw that in the chat box. Either gene E, F, G, H, or I, or J. See what we got. Got a ton of E's here. Got a ton of E's. Let's see. I got an H there, a G. All right. So let's see. Who's going to get the... All right, Donovan. It looks like you're the winner here. We've got gene G is the correct answer. All right. So why is gene G the correct answer? Does the liver, does the heart, does the brain, does the kidney, does the pancreas, the skeletal muscle, do all of them have to go through glycolysis and they need ATP? The answer is yes. So gene G is the correct answer because it's the only one that is present in all the different tissues, and all the different tissues definitely have to have glycolysis. So a lot of you that picked E, just because those three were dark and high amounts, that's not what we need to focus on. G is the one that produces it everywhere. Okay, so we'll finish up maybe with this question right here. All right. So the researcher observed that tissue was with a high level of gene H, messenger RNA, did not always give us gene H. So what would be a reasoning? You might have to type more than a couple words on this one. What would be reasoning to explain how tissue has a bunch of gene H messenger RNA, but we didn't have any gene H protein? So what could have happened to the gene H messenger RNA that there wasn't any gene H protein? So I know this is a crossover question, but this is good to integrate multiple units that you've already had lessons over. All right, see if we can throw that in there in the chat box real quick. All right, you guys have done a terrific job putting stuff in there. Let's see if we can finish strong on this. Okay. So I think you guys are making a very common error that a lot of people make. We don't need to go directly to mutation. When I read the essays of kids from all throughout the nation, when we say something doesn't work, we automatically go to a mutation. That's not where our brain needs to go. We need to think about good biology. Um, what happened? We know there's different ways that we get expression, different gene expression models. So if we got the messenger RNA, something had to happen between transcription and translation. That translation did not work, but transcription did. So one of those things could have been post-transcriptional modification. Like RNA interference, or we could have spliced it up and it could have been part of an exon. And we actually didn't come out of the nucleus. Any of those things. So mutation is not necessarily the best answer. A post-transcriptional modification or post-transcriptional expression change would have been a better answer to that. Okay, so I'm glad I put that on there so we could get that taken care of real quick. Okay. So we're getting close to an hour real quick here, Lou. I do have a couple of questions left, but I'm not sure that we're going to get to those.
You get about 5 minutes if you want. I think people are hanging in there, Robert, and you're doing a great job. So I apologize about the polling problem, but
no, it's
you're doing
okay.
a great job with the chat box. Thank you, everyone.
Yeah, I think the chat box is working terrific. Everybody's throwing a great answer in there. Okay. Well, let's look at this one, and then I've got one last thing to finish up with to just show you where you can practice some of these questions. Okay, we're going to get describe here. So the keyword here is, the keywords are always going to be bold. And one thing I kind of give a clue to say is we can mark out the words when we do them. So that makes sure we've done the whole question. So if we do describe and we mark it out, this whole question should be marked out by the time we're done. That way we know we've answered all the questions. Okay, so we've got these different organisms, penguins and two different kinds of seals here. We're measuring the time of their dive. So that's going to be the independent variable, and we're measuring their blood lactose levels as a dependent variable as over that dive. So describe the most likely shape of the curve if blood oxygen levels of the Weddell seals were plotted rather than blood lactate levels. So we want to, you're not going to have to make a graph, but what would it look like? What would it look like if we plotted oxygen levels of these seals right here instead of lactate levels? So you're going to have to give me a few words here. What would that curve look like? We've got a positive increase of that lactate right here. Instead of lactate, if I said oxygen, okay? I'll give you about 45 seconds to see what we can come up with there. Okay, so most of you are saying decrease. What I really like though is when you say it's going to start high, because we end up having at the beginning of that dive, we have a ton of oxygen, and over time we start running out of oxygen, which would mean we have to go through fermentation, and that's why we have an increase in lactate levels. So that is your hypothesis to explain the change in lactate levels over the 20 minutes. Okay. So that is my last question that I have. This is an old question that teachers on here that you might be able to throw to your kids as a review for today's session. What I want to show you real quick with this and finish, we have to make sure we describe one contribution of each of the following in ATP synthesis. So if we just say we break down a glucose, and the glucose gives us, let's say it gives us what? It's going to give us an NADH. Is that really ATP synthesis? Is that really telling me how that NADH is going to give me ATP? No. you have to make sure you answer the question as it's written. So it's asking you, what is this? What is glycolysis and pyruvate oxidation giving us in ATP synthesis? So you're going to have to say that NADH is later used in the electron transport chain to give us ATP. So that's a very subtle difference, but it's a definite difference in getting a point or not getting a point. So just for the recording, there's the answers that you would see if you want to go back and look at this later in practice. All right, on your AP classroom, under assessments, you've got these optional student practices that are marked with this little weird green bullseye. One of those optional student practices gives you a question that I told you earlier: describe, identify, describe, calculate. You can still calculate anything in 1 through 6. Math questions are still options. You can type those answers in. So anything you can type in, they can ask you. And there's another: predict, justify, explain. That is a good cellular energy practice question that everybody in this classroom has access to right now under that optional student practice tab in your AP classroom. So you can go practice that, and your teachers can go over what your expectation is on the actual the answers, and see where you're progressing. So we've got a little less than a month before we take this, and I know you're going to buckle down and continue to work hard and continue to study. And I appreciate over 8 of you being in here today. Hopefully it was a productive hour, and it was helpful in your studies. So if there's any questions, I'll be happy to stay on, but I appreciate what you've done today and your participation specifically in that chat with some very good answers.
And thank you, students, and thank you, Robert, and thank you, teachers who joined us. And feel free to send questions to me, and I'll get them to Robert. But otherwise, enjoy the afternoon, and good luck on your exams and your exam preparation.